Hi and welcome to today's webcast by Spago Nanomedical, where we will have Mats Hansen, the CEO, that will present during approximately 15 minutes. And after that, we will have a little Q&A session. And if you have a question, feel free to ask them on the right-hand side where we have a form that you can enter your questions. You could do it either in Swedish or in English, but this presentation will be in English if there's any English-speaking people that are interested. And with those words, I'll leave it up to you, Mats.
Thank you very much, moderator, and a warm welcome to all attendees to this webcast, which is prompted by the press release that we issued this morning regarding positive interim data in the SPAGO-PIX clinical trial. I will start to provide a brief background to what we are doing and to the project, and go through what we have seen so far. And after that, I will leave the field open for questions. So with that, I would like to start to provide a very brief background to Spago Nanomedical. We see ourselves as nanomedical experts, and we have developed a proprietary platform technology with particles that we can get to accumulate in solid tumors. This platform can be used for both diagnostics and for therapy. The portfolio of projects that we have is rather unique. We have solid proof of concept data in the preclinic setting, and with today's news, we also have indications of clinical validation in humans. And the clinical trial that we are running contains early efficacy endpoints, which also contributes to a de-risk development program. We are targeting major indications in both the diagnostic and the therapeutic setting. And we believe there is a continued high unmet clinical need for both diagnostics and therapeutics in the field of especially late-stage cancers. And we see that with this, both projects have rather substantial market opportunities should they finally get to the market. A little bit more about the platform technology. So what we are doing and relying on is physiological targeting of nanoparticles. We have developed a polymeric material that we can tailor to accumulate in solid tumor by means of the so-called EPR effect. This effect means that the blood vessels surrounding solid tumors are leaky and allows for macromolecules to pass out through the capillaries and into the tumor tissue. This effect has been confirmed in several different cancer forms, major cancer forms, including breast and colorectal and so on. And is also a prerequisite, I would say, for many macromolecular therapeutic agents. This efficacy is independent of drug release. That's very important to point out. So what we have are functional nanoparticles. By that, I mean nanoparticles that in themselves can execute what we want them to execute in the tumor. And we can use these particles to bind therapeutic radioisotopes, or we can use them as diagnostics when binding diagnostic elements such as manganese that we are using in SPAGO-PIX. We can use this platform to generate a means of optimized treatment in the sense that we can tailor the treatment based on the imaging in the actual tumors. And we believe that the milestones we have achieved so far, especially within the diagnostics side, supports clinical use of the platform material. And we also see that this is actually de-risking the development of the therapeutic project, because we can use imaging to pre-select patients for clinical trials, for instance, or for therapy later on. So if we go more into the diagnostic side and to SPAGO-PIX, we have started by focusing on breast cancer, because we believe this is an indication where there is a huge need for improved MRI diagnostics or MRI imaging. We know that the MRI is a very good modality to characterize the breast tumor. And we also know that MRI is already today used extensively in these patients. We also know that there is a problem today with a large fraction of false positive diagnosis, meaning that you believe there is something on the MRI images which is supposed to be cancer, but it's actually not. And there is also regulatory concern over the current contrast agents because they all contain gadolinium, which is an element that we should not normally have in our bodies, and has proven to be able to generate some quite nasty side effects as well. With this, we believe that a specialized product focusing on tumor imaging can be priced accordingly at the premium, which makes the addressable market for a product like SPAGO-PIX rather attractive. And we also see the potential beforehand to expand the use in other solid tumors, and now also in liver, or more importantly, pancreatic MRI. I will come back to this later. So all in all, we believe that serving the major clinical needs out there is something that we can address with SPAGO-PIX. So the SPAGO-PIX trial that we are currently conducting is done in order to demonstrate, first of all, clinical safety of the particles, but also imaging feasibility for MRI, more exactly contrast enhancement of solid tumors, in this case, breast cancer tumors. The study is ongoing at 2 centers here in Sweden, and the recruitment of patients with confirmed breast cancer was done in a first cohort that was completed early this year. And now today we issued a press release about the second dose cohort analysis that was completed recently. Safety is the primary endpoint in the protocol, but obviously MRI of breast, liver, and pancreas are important secondary endpoints to demonstrate clinical feasibility. And all in all, the results from this study is aimed towards strengthening the dialogue with potential partners for an out-licensing of this project. So getting into the data that we have seen, first of all, I should say that we have had a meeting with the Safety Review Committee in the study, and they have concluded that safety continue to look okay, and that there are no alarming safety signals preventing, so to say, continuation of the study. Even more importantly, we now see good images of breast cancer tumors. And I've added one example here. You can see to the far left an MRI image without contrast. So we do baseline imaging without contrast in all patients, and then we follow up with 2 time points following administration of SPAGO-PIX. And we can see at the baseline image denoted by the red arrow, there is a huge lump in the breast, which after 2 hours post-infusion of SPAGO-PIX lightens up. And I will try to point here with my pointer. You can see the rim of the tumor very well. You can also see that it's lighter at the edges of the tumor and darker inside. This is probably a sign of necrosis inside the tumor. So all in all, this image is very impressive in the eyes of a radiologist because it provides information not only on the exact, so to say, spatial location and constitution of the tumor, but also on the internal of the tumor and the potential aggressiveness of the tumor. So with this, I would say that we have a first validation of our platform technology in humans. It's very delighting, of course, for us. We see that this has a potential to be clinically relevant in breast cancer, which is very promising, of course. And we now continue the study at the current dose in order to collect more data on this dose level and to kind of build the data set for future use and future discussions. So this has not only implications on SPAGO-PIX, but also on our second project, TumorAD, because that project is also based on accumulation of our nanomaterial in solid tumors. I will come back a little bit to that as well. One interesting observation that we have made during the course of the study, and this goes actually for both dose groups, is that we have found excellent contrast in pancreas. We did expect to get a positive contrast in the liver because that's where parts of the particles and the manganese goes out of the body. We have not seen preclinical results where the pancreas has been so clearly enhanced as we have seen in the clinic. And this is an area that we have started to investigate, and we do see that there is a large clinical need for pancreas-specific contrast agents for MRI, especially since you want to follow patients who have cysts or precancerous lesions that may develop into pancreas cancer later on. So we see this as a very attractive opportunity to strengthen the package and for licensing discussions and potentially also for future clinical trials. I would like to say a few words about TumorAD as well, because as I said before, the findings that we have in this trial that our nanomaterial is actually accumulating in solid tumors in humans has a huge impact on TumorAD because it provides kind of a preliminary validation also of this project. So the TumorAD is based on the same platform material as SPAGO-PIX. We have tailored nanoparticles to get them to accumulate in tumors, and they are carrying a radioisotope, lutetium-177. With that, we have created functional nanoparticles that can be used to irradiate tumors from inside the body and offer a potential treatment for aggressive or metastatic cancer, which cannot be treated with the medicines of today. We recently appointed the candidate drug in this project, and we are now moving rapidly towards the clinic also here. And we believe this is a very interesting commercial potential in the field of radionuclide therapy. We've seen very attractive benchmarks that are on the market, and we do see an increased interest in this field also from the big pharma companies out there. A kind of rough estimate of the addressable market gives at hand around 145 billion euros, and with that you can calculate about sales potential in different ways. We have landed somewhere around 1.5 to 4 billion euros annually. So it's certainly an attractive market, and there is a huge clinical need, obviously, as you see in these rather large cancer indications that are listed here. So with that, I'd like to wrap up this short presentation to say that we believe that Spago is now for real an emerging nanomedicine leader, especially within radionuclide therapy, but also within tumor selective MRI diagnostics. We do have an optimized nanotheranostic platform where we can tailor our polymeric material and integrate diagnostics and therapy in order to personalize treatment later on. And with this, we also believe that the development is de-risked because we can use tumor-ad particles, for instance, to pre-select patients that are likely responders. Both products, of course, are first in class and proprietary, and we believe that this is a promising path to personalized treatment. So with that, I leave the field open for questions. Moderator, if you would be so kind.
Yes, thank you very much for the presentation, Mats. And if you have a question, feel free to ask them, and you do so by using the form on the right-hand side. And you can ask them either in Swedish or in English, and I'll bring it up to Mats here. And we have received several questions here, and I'll start with a few from Ludwig Svensson at Redeye. You write that you will continue to dose patients at this dose level. When do you plan to start dosing the third dose group?
Well, a very good question, Ludwig. Thank you. What we have decided was that we wanted to continue a bit on this dose level in order to build a more extensive database, both in terms of safety data, but more importantly, also MRI images. The images that we have gotten so far is very good, and we do have objective contrast enhancement in all patients of the second dose group. So we'd like to build on that a little bit more to kind of strengthen the package even more. Having said that, what we are targeting right now is to add an additional 3 patients on the same dose level to match the first dose level, which also was 6 patients, and then make a decision whether we want to actually escalate the dose. There are pros and cons about that, or we could even try to de-escalate the dose to see if we can get objective contrast at an intermediate dose level. So we leave both of those options open for the coming analysis, and we will obviously be transparent about which way we go later on.
Thank you. We'll continue with another question from Ludwig Svensson at Redeye. Is the hypothesis that a higher dose from these levels can contribute to an even clearer MRI contrast?
It could be. What we do see is, as I said, we have very good images already now. An increased dose could, of course, or potentially give even better images. We don't know that. And we'll just have to see. What we do, I mean, there's always the potential in a clinical trial that you run into safety issues when you escalate the dose too much. So we will have to see about that. But right now, I think the dose level that we are on is actually good enough. So I would say we'll see what happens in the next step. We might very well escalate to an even higher dose.
Thanks for that. Final question from Ludwig Svensson at Redeye. What does clear contrast mean in more specific terms? Will we be able to take part in more concrete data when the entire study is fully recruited?
Well, I've provided some insight here already in the slides I've shown, and I believe that the image I showed speaks for itself, as it certainly does in the eyes of a radiologist. Obviously, we will communicate much more when we have the full data set of the study. But I think we, with confidence, can say that we have very good images right now.
Thank you. Next question is from Magnus Bernett at Direct News. Today's press release about the study with SPOTR-PICS mentioned that SPA Nanomedical assesses that the study can be completed within reasonable time with the current centers. And if you can give more clarity in when the study is expected to be completed. So what is completion within reasonable time, so to say?
Okay, thank you, Magnus, for that question as well. Very good. Yes, we have decided, as you all know, the recruitment rate has been rather slow to this trial. It's and the COVID situation certainly did not help. What we have seen after the summer is that has been a much larger flow of patients coming into the clinic, and we believe that that will help us to be able to complete the trial within a reasonable time. Exactly what reasonable time means is, as I'm sure you can understand, very difficult to tell. And me, like everyone else right now, would love to be able to look into the future and see what's going to happen in society. But I can just conclude that we do see increased numbers of patients coming to the clinic compared with before, and we do hope that completion of the trial will go quickly.
Okay, thank you for that. Next question is from Johan. Are there any major players who have shown interests in the results?
Well, thank you, Johan, for the question. We have had discussions from before with the major players in the contrast agent field. The kind of general and collective message that we got from them was that they wanted to see clinical data. We did not have contact with them again based on the release today, but we certainly will, of course.
Yeah, and another question in the similar space, how much data do you need before you can approach big pharma, you would say?
Well, we believe that we need solid clinical proof of concept, and I think we are closing in on that.
And the next question is from Staffan Dittmer. What happens to the nanoparticles in the body after treatment? Do they remain, or do they break down, or what happens to them?
Thank you, Stefan. Good question. The nanoparticles leave the body within, I would say with SPAGO-PICS, we have rather small particles of around 5 to 6 nanometers, and they leave the body within most of them within a day, and the rest within a few days. Tumorad particles will stay a little bit longer because they are larger particles, around 30 nanometers, but I think I can already now say that we have seen almost none or very little toxicity of the particles themselves. The toxicity that we have seen in preclinical trials is derived from manganese in SPAGO-PICS and from the radioisotope in Tumorad, not from the nanomaterial itself.
Okay, next question is from Eva Rede, and it's about if you can say anything about when the clinical trial will start with Tumorad.
Yes, thank you, Eva. Good question. Well, as I said, we are progressing Tumorad as quickly as we can towards the clinic right now. We do target to be able to complete GMP production during next year, and also the preclinical studies that will be needed for a clinical trial application, and then hopefully we'll be able to submit the clinical trial application, let's say within a year or so, a little bit more than a year maybe.
Okay, final question here comes from Axel, and what will be the next logical step after the clinical study is done?
Well, for Spago Pix, we have been pretty clear that we do want to partner the project or out-license the project to a major player in order to set the best stage for the larger phase 2 and phase 3 trials going forward. So that will be our focus from now on.
Thank you very much for all the answers, Mats. And if you feel that you want to ask a question to Mats, you can still fill in the form, and we will receive them and we'll pass it on to Mats or someone else at Spago Nanomedical, and they'll return to you by email then. But thank you very much for the time to be here with us today, Mats.
Thank you very much, and thank you to all you who have asked questions and who have listened to this presentation. Thank you very much.